Let me begin. Hi, welcome everyone. Thank you for joining. Could you confirm if you can see my screen and am I audible?
Yes, madam.
Yes. Thank you so much. Okay. Yeah, I will begin. So this is the second problem solving session for biochemistry course, which is offered by NPTEL. And if you were there in my previous session, you would know that I have a set of like questions, some slides on the theory. It's more of a discussion. And today also I have the same type of patterns. But before that, I will begin with some parts of previous session so that there were few doubts in the previous session. And because this session is recorded and I will be uploading them later, for those students who had this down in the last session and could not join today, they might want to access the video later. So I will still discuss them. And then we'll move on with this week's parts. Okay. So if you remember, if you were there in the last session, you would have seen that we had discussed this question, which was saying that out of these given statements, which statements are true regarding carbohydrate? And we had this one point, which was the fourth point, which was about reducing sugars are capable of reducing ferric or cupric ions. And one of us had asked the question that what does reducing sugar actually mean? And I said that I'll come back with a more concrete answer to that. So like I mentioned, and like one of the person also mentioned in the previous session, the reducing sugars are basically those sugars which have free aldehyde or ketone group. So reducing sugar is basically referring to some sugar which can reduce any other component, right? Reducing agents. So for reducing is basically it can donate a proton. So that can only be done if it has a free aldehyde group. If it can also be possible if it has a ketone group, but the ketone has to undergo tautomerization to form aldose. So to form an aldose group. So basically a ketoenol tautomerization. So this O group here becomes OH. And this OH, this H can then be donated to reduce the other sugar or other component. So this, as we had the question were about the carbohydrates in general. So reducing sugar tests can be done using Fehling or Benedict test. And if you look at the theory of these two tests, this gives you the idea that what reducing sugar can do. So the principle of both the tests are the same. So these basically reduce the copper sulfate, so Cu2 plus oxygen state, to a Cu plus oxidation state. So you basically, let's say this is a test for identifying that the sugar that you have in your solution, is it a reducing agent nature? So you use this solution, Fehling solution. So you have the Cu2 plus oxygen state here and you have your sugar here. And then what you want to find out is if it forms a red precipitate. If it forms a red precipitate, what has happened is the aldehyde has, so it has reduced the copper Cu2 plus ion to a red precipitate Cu2O. And this red color will indicate that whatever sugar you had in your solution was of a reducing nature. Similar principle is also applied in Benedict's test. So when we say reducing sugar, what we mean is a sugar with a reducing capability. Okay. So well, this was one of the question. If the person is still on the chat and if it's not clear, please do let me know. But I'll move on to the next set of questions. So now that I've explained you this little part, for people in the chat, can you answer this question? This will explain if you have understood the previous part. So the question is, what are the oxidation states of Cu in Fehling's reagent before and after reaction with acetone? So if I put acetone and Fehling's reagent together, will the copper ion state change, oxygen state change?
Yes, 2 and 1, madam. Last option.
It says, okay, someone says 2 and 1. Okay, anyone else has any other answer to it? Maybe I'll wait for a few more seconds. I am on the chat screen, so you can also type your answers. I can see them.
I'm 2 and 4.
Okay. 2 and 4, 2 and 1. 2 and 2. Okay. 2 and 4. Okay. 2 and 4 answer. Okay. Anyone else? So copper in this Fehling's reagent has the +2 oxygen state. So the first point is, first part is obviously 2. So it is 2 and 2 or is it 2 and 1? All right, let me give you the answer. It's 2 and 2, which means the copper state will not change. That's because acetone is this ketone group. Ketone group does not reduce. It needs a like a hydrogen, a proton with itself. Now, I just told you in the previous slide here that ketone can undergo tautomerization. But when you talk about acetone, know that the tautomerization or autotautomerization of acetone is very rare in nature. So whenever you find acetone in nature, it's always in the ketone form. And because it is in the ketone form, it will not reduce. It has to have a proton to donate so that it can reduce the subsequent ion, metal ion. So it will be, the answer is 2 and 2. It means basically the oxidation state will not change if you react Felling's reagent and acetone. Did that make sense? Let me know if it did not make sense. Okay, I'm assuming that it is clear. Let me know whenever I can come back to this slide anytime you want. So that was the session question from last session. We have no more questions on that topic, but let's move on to this week's topic, which is in continuation with the previous week's last few topics that we had discussed. So this topic that we had already discussed in the previous session, which is called ionization of water, and this is what is one of the major component of your this week's lecture notes as well. So this one we have already done in the last week. So let's do it once again. So water has a slight tendency to undergo reversible ionization to yield hydrogen and hydroxide. So when you see hydrogen or a water molecule, it has this tendency to dissociate into H plus and OH minus ions, right? But this H plus ions, which is formed, it doesn't stay as an H plus ions, but rather goes and immediately hydrates the next water molecule, and that H plus becomes a H3O plus. So you can imagine in this chain, if this hydrogen molecule, so the proton from this hydrogen, which is the H plus, will hop on to the next hydrogen molecule. That H dissipated will move to the next. So it's basically a proton is hopping from one molecule to the other molecule. And this is important because with this hopping, what happens is that, so you can see here, the short hops of proton between a series of H-bonded water molecule affects an extremely rapid net movement of proton over a long distance. And this is much faster than true diffusion. So this is a way of diffusion, but it is a very fast diffusion if you compare it to other modes of diffusion. If you have water mediate diffusion, it's very high, and that's because your proton is hopping from one water molecule to the other. Now this whole dissociation, we can look at the equilibrium for this reaction as well. Now this is on the left is what is a standard equilibrium constant formula, where you have products and reactants. So your equilibrium constant is basically given by the reactants concentration divided by the products concentration. So now if we bring it to the water dissociation equation, which is water split into H plus and OH minus, the equilibrium constant here will be K equilibrium, H plus OH minus, which are the products, and the H2O, which is the reactant. Now, here, H2O at 25 degrees centigrade, if you have pure water, the concentration of the water is 55.5 molar. We will determine this value in a numerical in some times. But for now, let's just go ahead with the number. So if you substitute this concentration of H2O by 55.5 molar for pure water at 25 degrees centigrade, you can then come to this equation, which is a very, very important equation for you people when you are looking into the syllabus for all the numericals. This reaction gives you the ion product of water, which is what? For pure water, if you take the concentration and multiply it by the equilibrium constant, this is equal to H plus plus OH minus, which is equal to the KW, right? Now, what you need to substitute here, we will substitute the K equilibrium value, which is the equilibrium constant. People have calculated the key equilibrium constant by electrical conductivity of pure water. Remember, we are dealing with pure water here. So pure water's equilibrium constant is 1.8 into 10 to the power minus 16, again at 25 degrees centigrade. So you are substituting this value here, and ultimately your ion product is 1 into 10 to the power minus 14. this value 1 into 10 to the power minus 14 is constant always. Ion product or water is a constant value. It's 1 into 10 to the power minus 14. Okay, that means your H and OH minus ions can always equate to 1 into 10 to the power minus 14 molar square. That is always constant. Now the question is 1.8 into 10 to the power minus 16. How did that value come? Till now, it was this formula clear? Is any step not clear here or any value? I will wait for 30 seconds for you to look into the slide. Okay, then I'm assuming that this part made sense to you. Now, you people did not ask me that why is this 1.8 into 10 to the power minus 16? How did this value come? So this value came from this equation, this whole value which was determined by the electrical conductivity. This is what the reaction or this is what the equation is where you can determine the value. So following the same principle that we have been doing, K, dissociation constant, equilibrium constant being H plus OH minus divided by H2O. Here we will now substitute H2O with 55.5 molar because we are dealing with 50 pure water. Now, if you look into this, this is the same that we have just determined. If you reverse the equation, if you say that you want to look into the disequilibrium constant using the ion product by the 55.5 molar concentration of pure water, then your equilibrium constant comes to 1.8 into 10 to the power minus 16. So this is what they had done in the experiment. This is the value that we have used here, which gives you the ion product of the water. So this you should remember that ion product of water is constant. Here is a thing that I have not yet discussed, but that I will come to when I go to the question which is corresponding to that one. So what you now need to remember, ion product of the water is constant. This gives you a very important thing to remember. Now at neutral pH, H plus and OH ion minus concentrations are same, are equal. So if you do that, if you substitute that value here, so instead of H plus and OH minus, if you write H plus squared, which is basically squared value of the H plus ions. That means your ion product is now only determined by two squared values of H plus ions. So to determine the H plus ion, you square root Kw, which is 1 into 10 to the power minus 14. Then it gives you that at neutral pH, the concentration for H plus and OH minus is 10 to the power minus 7 molar. Equal. H plus equal OH minus. That is 10 to the power minus 7. Together, they make up the ion product, which is 1 into 10 to the power minus 14 molar square. Let me know if nothing was clear or if you have any doubt here. So let me again brief you through it. What we are trying to determine is we are trying to determine how does the ionization of water look like at equilibrium. So you have H2O splitting into H plus and OH minus. This formula here, equilibrium constant, this is a known formula where you look into the concentration of your product divided by concentration of reactants. Concentration of reactant, which is the water here, in pure water at 25 degree, the concentration is 55.5 molar. We substituted this value, cross multiplied. We got 55.5 into the equilibrium constant, which is equal to the concentration of H plus and multiplied by concentration of O minus, which is basically the ion, both the ions, so ion product of water. Now you need to substitute the equilibrium constant here, which we know by previous experiments that it is 1.8 into 10 to the power minus 16. We substituted that value, and that gave us that the ion product is 1 into 10 to the power minus 14. This value, I'm again repeating, this is a constant value. This is always the same. Now, you can play along with this. Now, you want to say that at neutral pH, when is pH neutral, when it has both the positive and the negative ions in the equal concentration. So using this idea, we now want to calculate at neutral pH what is the ion product. We know the ion product. what is the concentration of both H plus and OH minus, what is the equal amount of both, which is 10 to the power minus 7. So you know that at neutral pH, H plus minus ions or the OH minus ions are both at 10 to the power minus 7 molar concentration. Okay, thank you. All right, so now we have calculated at neutral pH what is the concentration. We'll come to a term which is pH. So how do you relate pH and H plus concentration? This is also very well known. Sorry. pH is basically log of 1 by H plus or minus log H plus. If you take H plus on the numerator, then it becomes minus log. So pH is basically minus log of H plus, right? Now, if you look into the same principle here, we have just said that H plus concentration is 10 to the power minus 7 at neutral pH. So if you want to find the numerical value of that pH, it will be log of 1 into 10 to the power minus 7, which when you finally calculate comes to 7. And we all know pH is equal to 7 is a neutral pH. So we have just used this value from our ion product equation, substituted that into the pH formula and found out the numerical value at which pH is neutral. And we have now proven that the pH 7 is neutral because it has both H plus and OH minus in equal concentration. It's not a difficult thing to understand. And on the right, this is from these slides are from no doubt, right? Okay. So the slides which I have made, I have used Leninger for reference. You can all refer to Leninger. If you have access to hard copy, then it's very good. If you have access to soft copy, even that is also okay. If you are for some reason not able to find the soft copy, please let me know. I can share the soft copies with you. So on the right is basically what they're trying to show here is at different pH, what are the general normal chemicals that show at that pH range. Right? So for example, gastric juices, lemon juice, they are highly acidic. But when you use bleach in your house or when you have household ammonia, those are increasingly or they're very basic in nature. While human blood, tear, these are neutral. They have a pH of 7. Okay, so now as previous people who've joined previous session would know, I have set of questions pertaining to this topic so that it helps you understand the topic much better. So let's begin with the first question. So here you have 4 sentences and 4 options. Let me know which one is the correct answer. So T is for true and F is for false. So the options are 1, 2, 3, 4, T, 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 F. So let me know which statements are correct and wrong. The statements are the neutral pH of water is independent of temperature. The solvation of hydrogen ions is mediated by hydrogen bond network. The unusual polarity, specific heat capacity, and surface tension of water are due to its hydrogen bond network. Fourth being the ionic product of water is constant. So which one of these do you think is false or true? And what is the correct answer in this question? I'm waiting here and I've already got one response. Maybe I'll wait for a few others and then we can discuss. Okay. All right. I think most of you have given the answer. Let me, and as most of you guessed it, it's FTT. All the statements are true. The last 3 being true because I've shown you that it's proton hopping. Hello, can you hear me?
No, ma'am. Yes, ma'am.
Am I audible? Okay, sure. Sorry, I just had a right. So, like I was saying, it was proton hopping that drives the properties of water molecules. The first one is wrong, and that's because I have been always mentioning that all the values that we have constituted or substituted in the formulas are all values of water at 25 degrees centigrade. If I change the water, the dissociation constant changes, the extent of dissociation can change, and that's why your entire values will change. So the neutral pH of water is dependent on temperature. The temperature that we are looking at pure water is at 25 degrees centigrade, right? And every one of you got right, so that's very good. Okay, next one. So which of the following molecule does not form hydrogen bond out of these four options? Is my voice better now? Am I audible?
Yes, madam.
Okay, now it's better, right? Cool. All right. Yeah, so the answer to the question, whichever I'm seeing people are writing, so let me just wait for them all to catch up. Hmm. Okay. I think people are still writing. Maybe I'll give the answer. We have a lot of numericals to do today. So many of you wrote many answers. The answer to the question is, and something that I have been mentioning always, please read the questions properly before answering. The question was about which of this molecule does not form hydrogen bond. So we are looking for a molecule that does not form hydrogen bond. And that's because if you look into the first three, HNO3 or H2O or HF, they are bonded by hydrogen bond, whereas in HCl, they are bonded by the sharing of electrons, so covalent bonds. So the answer to this is HCl. Okay. Next. This is what I had mentioned. How did we come to the value of 55.5 molar? So the answer I can reveal, but can you attempt to work it out? Like, could you think of why it will be 55.5 molar? It's okay if you can't. I will wait for maybe 30 seconds if you ever want to give it a try. And after that, I will discuss the answers. Okay. All right. People are giving me answers, and I think some of them are trying to solve it. Few more seconds, and then we'll do the this thing. Yes, I think someone has already got it. So I will wait for people, maybe. Okay, all right. Let me give you the answer. So like I said, it's 55.5 molar. That's the molarity of pure water at 25. And this is how we are determining it. Someone already told it's 1000 by 18. So let me tell you what we actually try to do here. So we are trying to find the molarity of pure water at 25 degrees centigrade, right? Molarity, if you remember, molarity is basically number of moles of solute given in a volume of solution in liter. So always remember the units which are in consideration. In molarity, we are looking at a volume of solution in liters, okay? And we know the density of water is 1 gram per ml, or it's equal to being 1 kg, 1 kilogram per liter. Basically dividing, multiplying both of these, numerator and denominator by 1000. 1 gram into 1000 is basically 1000 gram. 1000 gram is 1 kg. 1000 ml is 1 liter. So density of water is 1 liter, 1 kg in 1 liter. So if we know the density, we can find out the mass. That's basically density into volume. So you have 1 kg per liter into 1 liter, which gives you 1 kg simple, or 1000 gram. So the mass of water in 1 liter solution is 1000 gram, right? So you now want to find, because you need to have the moles of the solute. Number of moles is basically mass of the solute divided by the molar mass of the solute. So the mass of the solute here will be 1000. Molar mass is basically H2O, which is H2, so 2, and O is 16, if you remember. So you add them up, it becomes, atomic number becomes 18. So molar mass is 18. 1000 by 18 is 55.5. So overall molarity is 55.5 divided by 1 liter, which is 55.56 molarity. And that's how we had used that value. Did that make sense? Yeah? Okay. I tried to put it as molarity, because we are talking about this thing here, number of moles in volume of solution. Can you tell me what is the formula of molality? Someone asked molality, right? Is anyone on the group aware of what is the formula of molality? Yes. So 1 kg solvent, that's right. So your solvent here becomes in mass grams, so kilograms. But when you're talking about molarity, remember all the solutions we keep saying 1 molar solution, 2 molar solution, that's by volume. So moles by volume. So number of moles in the volume of the solution, which is in liter. Okay. Okay, next set of questions. These are all questions that will give you a good practice of understanding the pH, and we'll also do subsequent topics in the coming slides. For now, the equation that we had just done for water. So what is the concentration of H plus in a solution of 0.1 molar NaOH? If you have a 0.1 molar NaOH solution, what is the concentration of H plus ions in that solution? I'll wait in the chat box. Okay. Think of which formula can you use here when I'm saying H plus and 0.1 molar NaOH. Basically, you have molarity and you have H plus. Okay. People are typing. I can wait for 30 more seconds. Okay. No worries. We will do it. We'll look at the answers together. This is what I was saying. We know the Kw. I told you that's always constant. NaOH has OH minus ions, and we know the value of OH minus ions. We can easily calculate the H plus ions. I know it was very easy when it didn't click, but that's okay. That's what the entire purpose of this session is. We will see a different ways to solve the question. So you have OH minus, you have ion product, which is always constant. So you basically just substitute these value, divide the ion product by the concentration of OH minus, you get the ion concentration for H plus, which is 10 to the power minus 13 molars. Okay. Let's look at the next question. Now the reverse of it. So if you have an H plus concentration of 1.3 to 10 to the power minus 4, then what is the concentration of OH minus? This should be easy now that you have already seen one question with that answer. You basically have to apply the same formula. So you have the values of OH minus ions this time, and you need to calculate the opposite. You need to calculate the values for H plus ions. So what do you think the answer is? Okay. 30 more seconds and then we look at the answers. Okay, I see people typing. Point 769 to 10 to the power minus 10. Okay, one answer. Someone else is typing. I'll just look into that answer. 7.7. Okay, sure. Same answers. So the answer, as people have already guessed, it's these. Like basically, one the people who wrote, they are correct. So the same formula, ion product, which is H plus ions into OH minus ions. We have to find out the value for OH minus ions. So we'll divide the ion product by H plus ion concentration, simply substituting it. 10 to the power minus 14. So you take minus 4 in the denominator, it becomes plus. Minus 14 plus 4 is minus 10. 1 by 1.3 is, as people have written, 0.7. So what I have done is I basically have just removed one decimal point, was 10 times to the right. So 1 point to the right. So I have substituted this as 7.7 into 10 to the power minus 11. What people have wrote in the chat box is basically this point here. So 0.77 into 10 to the power minus 10. But they are basically the same answer. So people got it right. So let's move on to the next important topic of this week, which was about dissociation constant of weak acid. Now, why weak acid and weak bases? Because strong acids or strong bases, basically first thing is acids, as we've already seen. Acids are basically proton donors, so they can give H plus. Bases are proton acceptors. They will accept proton. Why? Because acids have H, as I've already discussed. Bases have minus negative charge on them. So to neutralize that negative charge, they will accept a proton to become stable. So bases are proton acceptors and acids are proton donors. Now, strong acids and strong bases, they are strong because they can completely either donate the proton or completely accept the proton, right? So they are very strong in nature. We'll see what strong we are talking about. What we are more interested in, weak acid and weak base, which do not ionize completely, which do not give away the protons completely. So what actually happens, how do they behave in a dynamic system is very interesting. And that's from there only we have understood the dissociation constant values and pKa values that are part of the next entire unit of this week's lecture. So acids are proton donors and bases are proton acceptor. Now, if you can see here, CH3COOH is basically acetic acid. So when acetic acids dissociate, it forms H plus and CH3CO or acetate and hydrogen ions, right? So a proton donor and its corresponding proton acceptor makes up a conjugate acid and a base pair. So you have a conjugate base for a conjugate acid. So in a acetic acid system, you have conjugate base, which is accepting the proton, and a conjugate acid, which is giving away the hydrogen, right? Proton. So this system is always there. Now, what you need to remember, and this is where it's very important for you to remember, the stronger the acid, the greater its tendency to lose proton. So if an acid is strong, it means it will give away its H plus to a very great extent, right? So that means it will dissociate very high at a very fast rate, that is large dissociation constant. So stronger acid lose H plus quickly, they have larger dissociation constant. Quickly in the sense they will have more H plus to dissipate. So larger dissociation constant. Weaker acid on the other hand have smaller dissociation constant, right? This you need to remember. The second thing that you need to remember is what is pKa. It's something that you will keep finding. So let's go back to this equation once again. This is what we have just seen in the previous part. So Ha is basically any acid can be written as H and the remaining base that it has. So Ha here in CH3COOH, which is acetic acid, H will be this part and this A will be CH3COO minus or CH3COO. So in any acid dissociates into H plus and A minus ions. And we know already this is how equilibrium constant looks. So this is Ka or dissociation constant. Ka is dissociation constant. So here, if you want to remember till here, the stronger the acid, the higher the Ka. Pk is basically taking log of 1 by Ka or minus log Ka. So pKa is basically minus log Ka. Greater the Ka, lower the pKa, lower the log value here, minus log. So how do you connect all of these? So stronger the acid, greater the Ka or greater the dissociation constant, lower the pKa. This relation you need to remember always. This is what is the most important relation here. Again, stronger acid dissociates, their tendency to lose proton is more. So larger dissociation constant, larger Ka. Larger Ka, if you, of course, if pK is being negative log of Ka means larger the Ka, lesser the pK. So strong acids will always have low pK, lowest pKa. Weaker the acid, more the pKa values, right? Is this clear? This here, again, you have this in your lecture notes as well. This is from Lenger. In this table, sorry, in this table here, we are looking into, or in this chart here, we are looking into different pH values, and we are trying to understand how they are related to Ks. So you have Ka value on your left under each of these acids or molecules, and the pKa value inside the figure. So you will see, let's say phosphoric acid, the dissociation constant is too high. So what should be the pKa value, high or low? You can take a guess. It will be low because Ka value is too high. And where do you see phosphoric acid? You see it here at a very low pKa value, which is also at low pH. Let's see an opposite. Acetic acid. The Ka value or the dissociation constant is very, very less, 1.74. So where on the spectrum should you find the acetic acid? pKa value. It will be higher. So where do you see pKa? You see them here. 4.76. This was 2.14. This is 4.76. Similarly, let's look at amino acid, glycine. Glycine has two such groups that can lose proton: carboxyl group and amino group. So carboxyl group and amino group, if you compare, Ka value of carboxyl is higher. So pKa of carboxyl should be low, which is here. Similarly, in the amino group has less Ka, so more pKa, so pK is here. So this is what we are trying to show visually here, that lesser the Ka, more is the pKa value, more the Ka, lesser the pKa value. Up until this, is this clear? Okay. So I am assuming that this part makes sense. Please feel free to unmute or put it in the chat box. I'll keep coming and checking. Now this you would have seen in the note. This is basically a titration curve, and that gives you the value of pKa of a certain acid or whichever molecule you're looking into, right? pKa value is basically you are so this is a titration curve. It's basically you are trying to determine the amount of acid in a given solution. And how are you doing this? You are using a known concentration strong base OH minus, so which comes basically from NaOH. So you use NaOH and you use, let's say, certain amount of acid that you want to find out how much amount is present in this particular solution. You put both of them, add both of them, mix both of them till they are neutralized. At the point that they're neutralized, you get to know how much of this acid was present in your solution. Now, what happens is you are having water already, and you have, let's say, your acetic acid, which is a titration curve for acetic acid. So what you're trying to do is you're adding the OH minus in small increments such that your H plus, so acetic acid is ionized at the beginning. What you are trying to do is you're neutralizing those acetate ions and the protons. So you're trying to dissociate the CH3OH as much as possible until you reach the end point at pH 7 where you can't see any more dissociation. But there will be a value which is when you have added 50, like 0.5 equivalent of NaOH, whatever you are using, whichever amount you're using, half of that base you have used. At that value, you know that the acid is equal to the amount of acetate ions that has basically the proton acceptor, the conjugate base, and the acid are in equal number. So that point is called the pKa value. When you have, so any reaction or any titration curve that you do, the value which you get at 0.5 of the NaOH, 0.5 equivalent of NaOH, is the pKa value for that amino acid. I explained this in short. If this is not clear, let me know. I will try to explain this in a bit more manner. If this is not clear. So we do the titration curve to find out the pKa value. How do you get the pKa value of any of the molecule that you're looking into? When you have added, you know you are adding base. You had, let's say you are using in a certain volume. The half of that volume of the entire reaction that you are using, when you have already, when you added that much base, what is the pH of the solution? That is what is the pKa. So you get the pKa or the dissociation constant of that particular compound or molecule that you're looking into, be it acid, be it amino acid, or be it any other acid that you're looking into. Any doubt? Okay. Okay. Now, few associated points with this. I understand this could be a bit, too much for understanding. I will again go slowly over this slide so that this is easier, and it will become easier as I go on in the next slides, hopefully. So let me again explain this to you. This is a titration curve for acetic acid. I'm trying to find out the pKa value. What I am doing is I am adding NaOH and I'm trying to find out the amount of acid in it. That is the idea of titration curve. Now, at the very beginning, I will have acetic acid in its form. Like it will not dissociate. It will dissociate, it will ionize, but not to a very great extent, right? Now, with increasing amount of OH, it will start to form acetate ions and protons. Acetate ions and protons. Remember, I also have water molecules which is dissociating. So I have my hydrogen ion going and attaching to other water molecules as well. So my H plus ions is being recycled and more of my CH3COH, which is basically my acetic acid, is getting ionized. Until I add 0.5 equivalent or half of what I am supposed to use for my base. I add that, and the pH of the solution at that point is where I know half of my acetic acid has already ionized, half is remaining. Half that has ionized is giving me the equal amount of acetic ions. So my acetic acid and my acetate ions are equal in concentration. At that point, this is what is called as pKa. So that pKa value is basically equal to the pH of the solution. And then you keep again further adding OH until every acetate ion dissociates. Every acetic acid has now become acetate ion. That is where ending start, and this happens at 7. Because you remember at 7, pH 7, neutral 7, your positive and your negative ions are in equal concentration. So your CH3COO minus and your H plus will be at equal concentration at pH 7. So that is where the ending is. Now, what is this blue mark bar here? This is called the buffering region. So basically here, if you add H plus or if you add OH minus, the buffer or the system will not see much effect on the, so pH will not see a greater extent of differences, right? In contrast, so if you add beyond 5.76, if you add, so you can see here, let's look at this point. If I add any more amount of OH, there is a sudden increase in the pH. Come here in this part. If I add more of OH, or if less of OH means more of H plus. If I add more of H plus, suddenly there is a drop. So this sudden jump is where you have sudden changes in pH. This zone in between is where the pH can still be in manageable condition. It does not show a very drastic effect. That range is in and around the pKa. So the pKa value is 4.76, 5.76 and 3.76. So what do you see? It's +1 and -1. That's the buffering zone of titration, buffering zone of acetic acid. You will find this buffering zone for all the biomolecules that you try to titrate. So whenever you look into a titration curve, you will see this shaded region, which is basically the buffer region, which is where if you keep at that pH, the pH will not change rapidly when you add H plus or OH minus ions. And that's why if people are there who work in lab, let's say you keep a solution as pH 8, you prefer to keep it at pH 8, but you can go up at 8.1, 8.2, or you can go a bit lower, 7.9 or 8.0. That's because at those concentration, the pH will not suddenly increase if you add H plus ions, or if it suddenly not decrease, it will still maintain the optimum pH until you put it at a very low value. Let's say 3.76 in case of acetic acid. You're making a solution. You're maintaining the pH as low at 3.75. If you add any H plus ions, or if it undergoes any changes, the pH of the solution will undergo a drastic change. So you have a buffer region for all the molecules that you can keep them in, ideally at pKa values. Okay. Now what the graph on the right shows? The graph on the right shows a similar pattern as this on the left, right? So you have your CH3OH here. But what is the difference? The difference is in the pKa value. And where do we get the pKa value? The higher the pKa value, the lower the dissociation constant. So they are weak acids. So as you go higher up, the weak molecules keep showing up. So your pKa value increases, your Ka value decreases, your Ka value decreases, it means these are weak acids. So the nature of the curve remains the same, but where is the curve present is where the difference lies. Right? So basically at 4.76, this molecule here has not even started to dissociate. It has dissociated only a bit to a certain amount. But if you look at the third component, which is ammonium ion, if you look at 4.76, it doesn't even care to dissociate. So different kind of molecule, different kind of dissociating tendencies. And that is reflected in where the position, the vertical position of the curve reveals how, to what extent the difference between the pKa value of different molecules are. But the way of reading is the same. Okay? Did that make sense? Any doubt here? Okay, if, for people here, the recording will already be available in the sheet that has been shared. So you can always go back and refer to these notes and videos. In case you have any doubt from the previous session, also you can reach to us. Right. So now let's move on to some questions pertaining to this topic. So the first is the pH of a buffer is calculated using which formula? There is a set equation that we use to calculate the pH. So do you people know which one is this out of these four options? Henderson, anyone else? This is easy. This is basically like someone said, Henderson-Hasselbalch equation. This is basically this. We always start with this equation for all the physical chemistry equation that we look for a product and reactants. So we know this already. What we are trying to do here is we are trying to find the pH. So pH is basically the value of H, the concentration of H. So let's just put right away that way. So let's put H plus on our left hand side. So Ka into H minus HA minus divided by A minus concentration. We are just basically rewriting the entire structuring of the formula. Now we will do logarithmic negative logarithm both sides. So the minus log H minus log K minus log HA by A minus. Minus log H plus, we all know concentration of H plus is basically the pH. So pH minus log Ka, we also know this. This is pKa. We have just done this in the previous class, previous slide. And this is minus log HA by A. Okay. So what we do is we remove this negative sign from here, put it plus. So if you're doing plus, you're basically inverting the equation. So you're putting plus log, but this time acid A minus by HA. So your equation basically gets reversed. So now if you want to actually look into it, you are basically saying pH is equal to pK, pK, how they are related. pH is basically pKa plus log of proton acceptor given proton donor. This is what your pH of a buffer is, how you are calculating. Now, interestingly, you will see, let's put it at this point here. Why are we saying pH is equal to pKa? Here, what did we say? That at this value, at the central or at the midpoint, what we know is the proton acceptor and the proton donor are in same concentration. So basically, these and these are same. So basically, it's 0. Log of 1 is 0. So that means this component completely cancels out. At your midpoint of titration, your pH is equal to pKa. So your pKa value will always be equal to pH in a titration curve. This is why at this point we write pH is equal to pK. So from this equation, from this curves, you not only find the pKa, that pK value is also equivalent to pH of this solution. Right? That was clear? Okay. Let's do more calculation so that it's better understood. So you have a pH of the solution, which is 5.8. What is the concentration of H plus? You can skip the units for the time being, but you have pH. What do you think the concentration of H plus will be? Let's do this calculation. All the different formula that we have just seen will make sense to us. You have H plus ions. You want to calculate the H plus ions, you have pH. And there is a very clean formula that connects pH and H plus ions. Okay, I see people are still attempting. So I will wait for a few more time. and then we will look into the answer, which is basically one simple formula, but it's better to try to attempt and then look into the answers. Yes. So let me tell you the answers. Like someone mentioned, 1.58 into 10 to the power minus 6. So the simple thing that you can understand is H plus is equal to 10 to the power minus pH. You don't have to remember anything else. 10 to the power minus pH. You have pH. 10 to the power minus pH is your H plus ion concentration. As simple as that. I said it's a very neat, clean formula. If you have used other formula of being pH having 1 minus log, that's also fine. That's also equal, and that's also your answer should come to 1.58 into 10 to the power minus 6 mole per liter. But this is one of the cleanest formula that you can use if you have H plus and pH minus questions. Next set of question. There is a mixture of acetic acid and sodium acetate, which is 0.2 molar and 0.3 molar. You need to calculate the pH of the medium, given that pKa of 4 is 4.676. The last formula, the last set of formula that we have joined pH, pKa, and the proton acceptor and proton donor. That is the formula that you need to use. Use that formula and try to come to the answer, and then we can discuss it. It's a straightforward question with all the values given. You just need to put the values in the formula, and you should be arriving at the answer easily. The formula, if people have not listened, once again, we looked into a formula which connected pH, pKa, and the proton acceptor and proton donor concentration. For your practice, this is just few set of questions that I have in slides. If you want to get a better grasp at these topics, there are many resources available on the net where you can access these questions. Try to attempt them, and that will definitely help your understanding and getting a grasp of these topics. There are a lot of numericals available on different resources online. Okay, I've received one answer. If people are still attempting, it's okay. I will reveal the answer. You can follow along and see if you were correct. So 4.94, as someone mentioned, correct. And this is the formula that we have used, as we have just mentioned, right? So the Henderson-Hasselbalch equation says pH is equal to pKa plus log of proton acceptor by proton donor. 4.76 plus log of proton acceptor here is sodium acetate. Remember acetate ions. So sodium acetate 0.3. Proton donor is the acetic acid. So the acid 0.2. So if you do that, is basically 1.5 here. So 4.76 plus log of 1.5, which if you calculate, you will get 0.18. If you add them up, it's 1.94. Okay. Let's do more of these type of questions so that it becomes more easier. All right. Third question here. Calculate the pKa of lactic acid given that when the concentration of lactic acid at 0.010 molar and the concentration of lactate. Remember the terms lactic acid, lactate. Try to identify which one is proton acceptor here and proton donor here. The lactate is 0.087. What is, and the pH is 4.8. So basically it's the same question, but this time you have to find pKa values. Easy questions. These are all easy questions. Simply looking into the formula, which shall be able to, you shall be able to answer it. The formula being pH is equal to pKa plus log of proton acceptor by proton donor. I'll wait for a few more answers this time because this is pretty simple, so it shouldn't be difficult. Let's all try to attempt it and try to get a better grasp at it. So these questions that you do numericals, they're not only from online sources or from the book. These are also questions from previous year's assignment, previous year's exam questions, some non-graded assignments, some graded assignments. So basically, the more you practice, the more you be able to solve these questions. And these are all mix of questions from this course, previous terms, or online resources. Okay. Received one answer. One more, and we go to the answers. Yeah. Okay. So people are giving me correct answer. The answer here is rounded off, but it's the same that you have written. So putting the same formula, pH is equal to pKa plus log of lactate, which is the proton acceptor, by lactic acid, which is the proton donor. So if you want to calculate for pKa, you take pKa on the left side, pH goes to the right, it becomes minus log. You substitute the values, you get 3.86, 3.866, 3.87, or round it up to 3.9. So great. Second last. Now you have so you calculate the pH for a mixture of 0.10 acetic acid, 0.2 molar sodium acetate, pKa 4.76. This time I will wait for the answer directly without explaining the question.
Hello, ma'am.
Yes.
I have one doubt, ma'am. Actually, in
Yes.
assignment one, they're giving the pH only, the solution of pH, pH, and asking the pKa directly, not giving any or acid or base molarity concentration.
There must be something else on the question as well. Are you sure you don't have any other values? Some other information? Let me check the assignment question once again. Give me a second.
Okay.
Yeah.
Directly they are asking that, just they're giving the pH only, then we are asking pKa like that, not giving
Okay.
any component of the buffer values, just
Okay.
giving the pH and asking the pKa.
Okay,
In
sure.
assignment
I will
one.
just take a look into it. Yeah, just give me a second. I'll take a look into it. In the meanwhile, you can try attempt this next question. I see a lot of answers in the chat box. Maybe
Okay.
I'll first reveal the question answer, and then I'll definitely come back to you. Give me a second.
Okay, ma'am. Thank you.
Like last time, this answer here is again rounded off, but I think people have got it correct, 5.06. So you have pKa 4.76, which is in the question. You have located your proton acceptor, proton donor. If you have done that, you get to this point, which is 4.76 plus 4.30, which is 5.06 or 5.1. Okay, this is same question in a different format. It says calculate the ratio of concentration of acetate and acetic acid required in a buffer system of pH 5.30. So acetic acid pKa we have been doing so far. If you haven't yet captured the pKa of this acetic acid is 4.76. So you have your pKa value, you have your pH. Now you need to calculate the ratio.
This type of question only, ma'am, in assignment 1.
Yeah. So I just mentioned that the pKa value of acetic acid from the questions that we have been solving, we know the pKa value. It's already in the theory as well, and we have already discussed it so far. So here we know the pKa value. You can use that information in the question. But let me just look into the question from the assignment once again. 'Yeah, people are still writing, so I will just wait. .5. Just a second. Okay, I see people answering the question. Okay, I see people have answered question, and this is what it is. I saw someone also writing 0.54 by 0.1. So let's see if you have your doubt clear here. So, so what you are supposed to find is this component here. You are given both the pH. We know the pKa from our experience. So we know that the log of acetate to acetic acid is 0.54. And what we are supposed to do is we are supposed to look into the value of the ratio. So ratio is basically if log of acetate by acetic acid is 0.54, acetate by acetic acid is just anti-log of 0.54, which is 3.5. So most of you got it right, and that's correct. Just a second. All right. So let me quickly move to the next part, which is also okay. Now here, like I always keep mentioning, these are, what do you say, inference based questions. So you read the questions, you have certain statements. Here you are supposed to tell me which ones are correct based on the table that is given to you. So there are some pKa values given for different kind of groups: acetic acid, glycine carboxylate, glycine amino, methylamine in protonated form. Now on the basis of these pKa values, you have these set of 8 statements. Read the statement correctly and then tell me which ones are correct and which ones are wrong. This will take a bit time. It's okay. You can take your time, sit here. I mean, you can look into the questions and let me know. And then we'll look into the answer properly together. I'm trying to look for the question that you said. Maybe I'm not able to find, but whatever I see on my screen has the information which you require to solve a question. Could you specifically tell me which question, if you are referring to one single question? Because if what I see on the screen is basically all the information are some way or the other given. Maybe I'm missing out the part. You can just let me know which questions you are referring to, or which assignment you are referring to. Yeah, this is a longer question, so I will wait till people take their guesses and then we can discuss the answers. And yeah, you can definitely share the question. We can see how we can help with it. Yes. One more minute and we can then discuss the answers. A, B, C, correct. Okay. G is correct. Okay. I see people have reached till G, so maybe I'll give the answers. And D and E are also correct. Okay. All right. D and E are also correct. All right. Let me give the answers. Okay. You can see the answers. The correct statements are B, D, G, and H. Okay. Yeah, I see most of you have got a bit confused. No worries. We will see it one at a go. Okay. Why all of the statements are correct or incorrect. First statement says order of acidic strength, glycine is lesser than acetic acid, which is lesser than glycine, which is basically lesser than methylamine. Let's look at the value of methylamine. 10.66. Glycine amino acid is 9.6. Acetic acid is 4.76 and this is 2.34. Remember the acidic strength. This is pKa values. How was the relation of pKa values to the acidic strength? Greater the pKa value, lesser the Ka, so lesser the acidic strength, less acidic solution, weakly acidic. So it's the opposite. Your pKa being lowest means your Ka is the highest. That means you have the greatest out of these lists, you have the strongest acidic property. So your oscillator of acidic strength is the reverse. Did you get confused or yeah? My cue, which is what I said in the last also, before attempting, read at least 3-4 times what is the question saying. The question is saying acidic strength. I see many of you have written A is correct. Some of you would have also thought about A correct. For those people, is this clear why A is incorrect? pKa value is basically an inverse of your acidic strength, right? Okay. Next. Any question? Next, the carboxylic group of glycine, which is this 2.34, is more acidic than that of acetic acid, which is 4.76, due to deuterion stabilization, which is correct. Correct, right? This is here, this is more acidic than this. Makes sense. But if this is correct, then this should also have been correct. That's okay. Let's see is the amino group of methylamine here is less basic than that of glycine due to deuterions stabilization when the latter gains proton. It is more basic because it is towards the basic value. Methylamine is 10.66, glycine is 9.6, so it is more basic, not less basic. Okay. D, in a solution of glycine at 2.34, there is equimolar mixture of Na3 plus CH2, so glycine, and the proton acceptor. So proton donor and proton acceptor are in equivalent concentration, right? Now, when you have a glycine at 9.6, which is basically the amino group, you have NH3 plus CO2 minus NH2 CO2 plus, which is incorrect because it has to have an acetate group still. So it has to still have a proton acceptor, which is not present here correctly, right? So that's why it's incorrect. Sixth, the titration curve of glycine against sodium hydroxide will have a lower number of equivalence point than that of acetic acid. I don't see anybody writing F is correct, which is okay, but the explanation is here. The explanation is acetic acid has only this COOH minus group, so only this will have a pKa value. Whereas if you see in glycine, it has a NH2 group and a COH group. So it has two such groups that can ionize, and that will give you the pKa value. So it has two values. And it said, the statement said glycine has lower number of equivalents than acetic acid, which is the opposite. Glycine has more number of acidic groups, so it has more number of equivalence point, right? So next is G. At 2.76 acetic acid, which is 2.76 is basically lower than the pK value. So it has only few of the amino acids, a few of the molecules that has undergone ionization. So it is 1% ionized. At 4.34, if you look at the carboxylic group, 2.34, it is at the higher end. So it having 99% ionized molecules or ionized carboxylic group makes sense. So this is correct. If G is correct, then H is also correct. Anybody has any doubt here? And where do we have these values from? If you go back once again, let me just give you once again the perspective here. This value. These values, the buffer zones is where it gives you the percent ionized. So percent ionized here is 1%, percent ionized here is 99, because beyond this, it will all of them will get ionized and the solution will become neutral at neutral pH. Okay. So that is where the question comes from. So you look at the pKa value and then you look at the values that are given and see at which end of the spectrum the value is, and does that make sense with 99 and 1% iodized. Any question in this part, please let me know. All right. You can always come back and let me know if you have any doubt. Okay. Next set of, again, inference-based question. These are all important questions for you to understand the concepts. So you have pKa values for these different bases, all the different DNA, RNA bases, adenine, guanine, cytosine, thymine, uracil. Your DNA is initially at a pH of 7. And then you add the bases, concentrated bases, and you have these 6 statements. So which of these following 6 statements is correct? Take your time. There's no rush. We'll do it easily. And then we'll see the answers to it. Okay, this seems to take a bit time. No worries. 7:28. Maybe I'll wait till 7:30 and then we can discuss the answers. The answers are not difficult, but it's again, I understand because it's based on the pKa values of the bases which you are still trying to guess. It's okay. It doesn't matter. If you're not able to answer till 7:30, we will look into the solutions together. BDC correct. Okay. BDC correct. Yeah. Okay. One more minute and then we discuss the answers. E is also correct. Yeah. Right. Let me give you the answers because we have to finish by 8. Don't know how many I will finish. Okay, these are the answers A, B, C, D, F. Okay, let me give you the answer one by one. So the first one says the DNA, the proton most susceptible to being ionized can be found in cytosine. So if you look into the uh, pKa value, or if you look into the structure of the this thing of this different groups, you can see that cytosine will be more susceptible to being ionized. So you can see that from the structure. Second, when the base is first added, the pH increases. Now this, I would like you to look into this table here. Now you see, so DNA is more stable at pH 7. Okay. Now, when you are adding your first base, the pH will increase. That means it will become basic. But the double stranded will remain close to 1. The fraction, in the sense, the DNA will remain in double stranded form. Nothing will structurally damage the DNA. It's only when the pH reaches a very strong condition, which is more than 9.59 or 10.5, it's when the DNA denatures due to protonation of bases. So the deprotonation, the removal of protons from the bases, it becomes more evident at very basic pH. That's why when you are adding the bases at the initial stages, even if the pH increases, the double-stranded DNA doesn't seem to be changed at all. Next, as the pH approaches 9.8, the fraction of double-stranded rapidly decreases. This is exactly what I said right now. As your pH keeps going higher, it reaches to a very great extent of basicity. Your bases start becoming deprotonated, and that leads to the rupturing of the hydrogen bond, let's say between cytosine and guanine, and that will lead to the disruption of the structure of DNA. Now, E says if an acid was added instead of base, so you are basically making it more acidic, the fraction of double-stranded DNA would not change. That is not correct, because if you make it slight, so look at here. Basically, if you are adding more acid, it's becoming acidic. Even if it is mildly acidic, DNA starts to get destabilized. It becomes destabilized at 5 or 6. If it goes to 4, it becomes denaturation again, loss of hydrogen bonding. If it is extremely acidic, it becomes complete degradation because of backbone breakage. So an acidic change to the pH makes more effect on the DNA than a basic change. That's why E is incorrect. And F is secondary structure in RNA will get denatured at a higher pH than DNA. That's because if you look into secondary structure, they have these loop structures. They have which are more how do you say? They're more susceptible to a damage. That's why they will be more affected. Okay. Yeah. Right. Next, very interesting question. Look into this. This is the last question of this section on pH and pKa. So on your right or column 1, on your left on a column 1, you have 3 bases or 3 amino acids. Sorry. So 3 amino acids which are titrated against with a base. The titration curves of these 3 amino acids are given on your column 2, but not in the similar pattern. So tell me which one refers to which. So if you take column 1, A, so which one in column 2 graph refers to or belongs to the first amino acid or first molecule? What you need to see is how many groups of deprotonation that molecule has and how many such equivalence points you find in the graph. Two, three, one. Okay. Anyone else? Or maybe I'll wait. Maybe try to answer. Okay, V3. Okay, V3. Okay, I'll wait. Maybe I will wait for you people. Okay. I will give the answers. You can try and see if you were thinking in the correct direction. The answer is 231 as someone mentioned. Someone also mentioned B being 3, which is also correct. And the explanation is here. So if you're looking into the structure and if you are aware of the amino acid structures, the first one is basically alanine, which has a neutral amino acid with no ionizable side chain. B is a arginine. So it has, after this, if you see a guanidinium group. And third is aspartate because it is aspartic acid or aspartate, which is basically having a COH group, right? Now let's look at the titration curve in the same manner. Let's look into the titration curve too. This has two pKa values and two pKa values, as we know from the two groups that it has, COH and NH2. It doesn't, it cannot, the CH3 cannot be ionized. So CH, one for COH group, one for NH2 group. So this one makes sense, A is 2. If you look at B curve 3, this has three pKa values. Now the buffering zone, if you look into this, is at a higher pH, which is characteristic of a basic amino acid like arginine. And let's compare this to the titration curve 1, which has again three values, but with an additional buffering at a lower pH. So lower pH for the COH group, while this one has a buffering at higher pH because of this guanidium group. And which most of you got it correct. So very interesting answers. And you can, at the same time, while you are studying, you can look into the other amino acids group titration curves as well and try to look into the similar kind of pattern if that makes sense to you. You can try to identify how many ionizable groups that amino acid has and how many such pK values can you point out on the graph. Right. So if you have looked into the week 2 lecture notes, you will see that after pH and pKa, we come to analytical methods, among which you would have looked into SDS page, you would have looked into Edmond degradation and chromatography. So in my slides today, we have SDS page and chromatography. We are left with 20 minutes, so I will try to see how much we can complete. At least we'll try to complete SDS page today, right? Okay. Or if time permits, we can also cover chromatography, at least the principle of it. Let's quickly look into the second topic of this week, which is SDS page. So we have looked into protein, we have understood protein structure, we have understood how the pH works, how everything works. Now what we are trying to understand here is how to look into these different kind of proteins and how to visualize them, how to work with them in the lab. So the very common and the very most used methods for such purpose is SDS page. And if you would have studied, if you would have looked into the notes, it's basically telling that you are trying to run different proteins on the gel, and then you are trying to resolve them based on their molecular weight. And basically, if you look into the full form here, SDS here is basically sodium dodecyl sulfate, which is basically a detergent. Page is polyacrylamide gel electrophoresis. So this is gel electrophoresis. Basically, you are giving electrical field in a gel so that your molecules move across the gel with respect to their molecular weight. The molecular weight will determine how fast the molecule travels in the gel. If the molecule is small, it will travel faster. If the molecule is bigger, it will travel slower. But other than molecular weight, for a protein, you also have its charge. Your charge can also affect how it's moving. So what you do in this electrophoresis, you put SDS, which is basically a detergent. So what it does, it gives a linear negative charge to the entire protein. So now your protein, irrespective of what charge it has inside, on the surface it is charged with a negative charged. On the surface is negatively charged. So the charge component is now neutral, same for all the protein molecules that you load onto the gel. What will now determine the resolution is the molecular weight. So when you then look into the molecular weight, when you then look into the pattern of gel electrophoresis, you can identify which sample had a larger molecular weight protein than the other one. And this is what you do. You basically, so SDS page has two layers. You have stacking gel and resolving gel. Stacking gel is basically where you are allowing your molecules. So this are wells, which you see here. You have several components that makes up this gel. As we move ahead in the questions, it will become clearer. So you have basically an acrylamide gel here. The first layer is called stacking gel, which basically you load your sample, allow it to stack and allow it to take its size. And then in the resolving gel, which is in the later part, it's where your molecular weight comes into play, and that separates out how based on its weight. Right? And because these are proteins, these are negatively charged, the direction of migration is positive to negative to positive, so they will move towards the positive terminal. Okay. So what is on the right? On the right is how it looks right after your run. So how do you visualize this? You use Kumasi blue stain, which binds to the protein, and then that each band on the gel will represent the protein because it would have bound to the Kumasi blue, and your Kumasi blue will give you where the protein is located on the gel. Okay. Now, all this is very technical. This is easier to understand conceptually. How do you use it? I just said that you can use it for understanding the molecular weight. So how do you do this? Every time you run a molecular component on a gel, the first lane should always be a ladder. What is a ladder? What is a marker? It is basically a set of bands with defined molecular weight. So let me explain. What this is, let's say a gel that you have run, first and the second one only. This is the marker or ladder. And on this ladder, you know that at this band here, the molecular weight is 2 lakh kilodalton. Here it is 116097. So you know it, and you can see it has also gradually decreased because obviously the larger the molecule, the higher, the lesser distance it has traveled. And this is from respective proteins of myosin, beta galactosidase. Now I have a protein which is somewhere between 45,000 and 31,000. Visually, I can tell that the protein molecular weight will be somewhere around 40,000, right? But there is a defined way, or is a good way of how you can determine the molecular weight. You can do that by taking the log of these molecular weight from the marker and the migration on the gel. The migration, as you can see, it can be determined by the length of the gel. So you know the length of the gel. Let's say it's 80 centimeter. You see set of markers here. Let's say 20,000 has only moved this much centimeter, 5 centimeter. 66 has moved, let's say 15 centimeter. So you know the relative migration, and you can find out the log of these values, plot it, get this curve, standard curve for your known marker, and then use your unknown marker's distance and then find out the molecular weight for the unknown protein. This sounds complicated, but it is not. This is exactly where we will use it. So let's try to see how much we have understood the concept here. So you have your 3 proteins here: protein A, protein B, protein C. Protein A, you know the molecular weight, which is 10 kilodalton, migrated 15 centimeter. Protein B, molecular weight is 100 kilodalton, migrated 5 centimeter. And protein C, which has migrated 10 centimeter. So you have molecular weight for A and B, and you have distance for protein C. Can you use this information to find out the molecular weight of protein C? What we told you is there's a plot here which is based on the log of the known molecular weights, and on the x-axis you have the migration distance basically, and you give a standard curve there, and then you find out the molecular weight of the unknown. So if you remember the equation for the linear curve, linear graph, and then you use it for calculation of unknown, this is what I'm going to use. Let's try to do this. This will be a good exercise for you to understand the concept. Maybe I think based on the timing, we can only complete SDS page today. So in the next class, we will continue from chromatography. But let's complete the SDS page complete today. So let me try to give you hints. So you have 100 kilodalton, 5 centimeter, 10 kilodalton, 15 centimeter. We're supposed to take log of the molecular weight. So you can take log of the 10 and log of 100, and you have the respective y values. So you have respective x values and respective y values. If you have 2 points of y, then you can find out the straight line equation. Can you come to the straight line equation first? If you do that, then you will be able to find out the values for the unknown y here as well. Okay, I will do the first question because the next there is another question based on similar lines. Let's do the first one so that the second one becomes easier. Okay, so the answer is 31.6. We'll see how. Like I said, you have protein A, which is 10 kilodalton, has moved 15 centimeters. So we have seen already x-axis is migration. So on x-axis, if there is a 15 centimeter migration, then the value on the MR or value on the y-axis is log of 10, log of the molecular weight. So log of 10 is 15 centimeters. That's 1 point on your graph. Second, 100 kilodalton has moved 5 centimeters. So if this x-axis is 5, y-axis has log of 100. So that is the second point. Now, if you do the values, log of 10 is 1, log of 100 is 2. So 1 and 15, that's your 1 point on the graph. 2 and 15 is your another point on the graph. So your 1 will be here, 15 will be here, so it will be up. 2 will be here, 5 will be here, so it will be bottom. So it will be a negative trend always, obviously, because it moves in the opposite way. The higher the molecular weight, the lesser distance it has traveled. Now, simple maths that we have used. If we have two values, we can use to look into the straight equation. So how do we do that? We do y minus y1 divided by x minus x1 is equal to y2 minus y1 divided by x2 minus x1. This is a standard linear graph equation. So y minus y1 divided by x minus x1, which is equal to y2, the two values that we used, y2 minus y1, because what we are assuming is that y minus y1, so any point y, let's say at any point y1, this minus this is equal to the other values also. So distance from here will be equal to the fraction of the fraction or the percentage or the ratio of the difference will be same in a straight line. So y minus y1 divided by x minus x1 is equal to y2 minus y1 divided by x2 minus x1. Y and x are our unknown for the unknown protein. But y1 and y2 we know. We can consider any of these as y1 and any of these as y2. Let's consider this set as y1 x1. So 1 and 15. Y2 as the other set, 2 and 5. So 2 and 5, 1 and 5. Then it's basically just opening the equation. So you have 10 here, minus 10. This goes on the left. 1 x minus 15 goes up. You have this equation minus 10y plus 10 is equal to x minus 15. We further solve it so that we get 10x plus 10y is equal to 10 plus 15, which is x plus 10y is equal to 25. Okay. Now, this is your equation. This is your straight line equation. X plus 10y is equal to 25. Then you use this unknown. You know x, the migration here is 10. So your x is 10. You just need to find out y. So your y comes out to be 1.5, which is correct because 10 is here. So your y should also be in the center. So y is 1.5, but y is actually log. So to find out the actual molecular weight, you need to do anti-log. Anti-log of 1.5 is 31.6, which is kilodalton. Any doubt in this entire flow? Okay, maybe we'll stop here today. Let's all try to attempt this one last question, and then we'll continue this from the next slide, which is again a last question on SDS page, but that's okay. We'll do that in the next session. But let's finish it here and let's try to understand it better here. Again, similarly, you have a 3.9 centimeter distance travel for 45,000 Dalton. You have 6 centimeter for a 14,400 kilton molecular weight. And then you have an unknown which is between somewhere here. So can you guess the molecular weight? Use the same. I will stay on this line, maybe you, or wherever you want. If you want any help, let me know. So you determine in your Dalton and then answer it in kilodaltons. Okay, kilodalton is basically that divided by 1,000, or that divided by 1,000. Yes. Same. This will be a good exercise for you to just revise what we have done in the previous this thing. If you want to look into the values, this is what we have done, and these are your values. So let me know if you want to go to the previous slide. We have some 7 minutes left. Let's just attempt this question, and we will do chromatography in the next session. Do the same way. X1, Y1. Take Y1 as log of the molecular weight. X2, Y2. Y2 as a log. Then you have X1, Y1, X2, Y2. Use the standard curve equation to find out the equation. And then substitute it with the X values that you know here to get to the Y. Circular, but takes time, but it gives you the proper understanding of what exactly we are trying to achieve with SGS page. And this is the same with aerogel electrophoresis as well, where you do it for DNA visualization. Okay. This is the answer. I will also show the answer so that you can track at which step you are currently. And we'll follow the next slide from the next session. I will explain to you in 1 minute what exactly we have done. But in the meanwhile, you can check if you had done the steps correctly. Okay, let me explain. So the similar idea here as well. You have your molecular weight 45,000, which has traveled for 3.9. So you take log of 45,000, which is 4.65, and x is 3.9. That is one value. You take log of 14,400, and that is for 6 centimeter. So 4.15 is for your 6 centimeter. That is a y2. You substitute in this formula y minus y1 divided by x minus x1 divided equal to y2 minus y1, x2 minus x1. We can take any set of the values as one set of y1 x1. So I took this. So y minus 4.5 divided by x minus 6, y2 minus y1, which is 4.65 minus 4.15, 3.9 minus 6. I get to a value. I solve the equation. The final equation comes out 0.5x plus 2.5y is equal to 11.715. Then I use this value here. X for my unknown is 5.8. Basically, I just substitute x with 5.8, derive the y, and again do the antilog for y, come to a value for the molecular weight, 15.848 Dalton. The question asked me to near it to a nearest integer for kilodalton. So I do divide by 1000, and then I get it 16 kilodalton. That's what I have done. It seems very difficult, but it is not. It just follows logic. But here, we stop here today because let's not do any more questions today. And we already are out of time as well. So if you have any doubt in today's session, please feel free to reach out to me. Post your questions in the discussion forum. If you can reach out to me personally as well. Let me know if you have any other doubts. If not, then we can meet in the next session. This slide and this entire recording today will be available on YouTube. You can refer to the videos and let me know if you have any problems regarding this. And yeah, if you have any other doubts, so please feel free to ask. I am here. If not, then we can meet in the next session. Thank you so much. Thank you.
Thank you, ma'am.
Thank you. Thank you so much. Thank you. Next Tuesday, same time, 6 to 8.